Assalamualaikum and hello everyone. My teammates consist of Iza Shafika, Avian and myself, Siti Nur Azihal, would like to present on research regarding on DNA metabarcoding in forensic entomology. DNA metabarcoding is a cost-effective species identification approach with great potential to assist entomological ecologists. It is a molecular technique that uses universal DNA barcode markers and high-throughput sequencing (HTS) to identify multiple species from complex or mixed samples simultaneously. Traditionally, forensic entomology depends on identifying insects based on their physical characteristics. However, this approach has several limitations. It requires highly trained taxonomic experts and becomes very difficult when samples are damaged, fragmented, or incomplete. Early life stages like eggs and larvae often look very similar, making the accurate identification challenging. This issue can affect the accuracy of postmortem interval estimation. To overcome these problems, molecular approaches are used. DNA barcoding identifies a single species from one specimen at a time, usually using single sequencing. In contrast, DNA metabarcoding allows multiple species to be identified simultaneously from mixed samples using next-generation sequencing. This makes metabarcoding more faster, more efficient, and more suitable for complex forensic samples. DNA metabarcoding targets specific genes for specific identification. The COI gene is commonly used because it provides high resolution at the species level. The 16S rRNA gene is also useful, especially when DNA is degraded. These genes are sequenced using next-generation sequencing (NGS) platforms such as Illumina, MiSeq, or Oxford Nanopore. The workflow of DNA metabarcoding begins with sample collection followed by DNA extraction. The target gene is then amplified using PCR and sequenced using next-generation sequencing. Finally, bioinformatics analysis is performed to generate a list of insect species present in the sample. This is the example of workflow procedure in PCR-based DNA metabarcoding, which is from sample collection until species identification. Overall, DNA metabarcoding provides a powerful alternative to traditional methods by enabling accurate species identification even in complex forensic conditions. These technologies set the foundation for advanced forensic application, which will be explained next.
Now let's look at how DNA metabarcoding improve forensic entomology analysis. First, it allows species identifications from mixed insect samples and remain effective even when specimens are degraded or fragmented. Second, it works across all insect life stages, including eggs and early larvae, which are difficult to identify morphologically. In addition, DNA metabarcoding can also be applied to environmental DNA, meaning insect DNA left behind in soil, clothing, or surrounding materials. Altogether, these advantages support more accurate postmortem interval estimation. Now I will explain these advantages in more detail, starting with how DNA metabarcoding helps analyze mixed and degraded insect samples. At real crime scenes, insect evidence is rarely clean or intact, and investigators often collect mixed samples containing multiple insect species, along with damaged larvae or fragments. Traditional morphological identifications becomes more extremely difficult under these conditions. Therefore, DNA metabarcoding overcomes these limitations by extracting DNA from the entire mixed samples and identifying all species present simultaneously. This approach improves accuracy, especially when physical characteristic is no longer visible making it highly valuable for forensic investigations involving decomposed remains. Another major advantage of DNA metabarcoding is the identifications of early insect life stages. These stages lack distinct morphological features even for trained entomologists. However, accurate species identifications at these early stages is important because different insect species develop at different rates, which directly affect postmortem interval estimations. By analyzing genetic markers like the COI gene, DNA metabarcodings allow reliable identifications regardless of the insect developmental stage. DNA metabarcoding also enables the analysis of environmental DNA or eDNA. This refers to the DNA traces left behind by the insect in materials such as soil, clothing, or the surrounding environment. In cases where insects are no longer present at the scene, eDNA can still provide valuable forensic information. This approach expands the scope of forensic entomology beyond direct insect collections and allows the investigator to reconstruct past insect activity at the crime scene. One of the most important forensic applications of DNA metabarcoding is postmortem interval estimation. Different insect species colonize a body at the different stages of decomposition, forming predictable succession patterns. DNA metabarcodings allow forensic scientists to detect multiple insect species from a single sample, which providing a more complete picture of insect successions. This improves the accuracy and the reliability of PMI estimations and support the traditional forensic entomology methods rather than replacing them. Overall, DNA metabarcoding enhances forensic entomology by enabling accurate insect identifications from mixed, degraded, and early-stage samples, which strengthening postmortem interval analysis and forensic interpretation.
Previously, my group members had presented on how metabarcoding works and how it improves forensic entomology in real cases. Now I'll talk about bioinformatics analysis, validation challenges, and future forensic use of this technology. So, after sequencing, DNA metabarcoding does not end in the laboratory. It relies majorly on bioinformatics, which transforms millions of raw DNA reads into useful forensic evidence. Without computational analysis, next-generation sequencing data would be unusable in forensic entomology. Next will be about bioinformatics pipelines process sequences, which uses either OTUs, operational taxonomic units, or the more recent one, which is ASVs, amplicon sequence variants. So between these two, ASVs offer higher resolution because they can differentiate DNA sequences down to a single nucleotide, which enhances the accuracy of species identification and improves reliability and reproducibility across different forensic laboratories. So, after sequences are processed, they will be compared with reference databases such as BOLT and GenBank. However, incomplete or misannotated databases remain a major limitation, especially for forensically important insect species that directly affects identification accuracy. Then, recent advances in automation and machine learning helps in minimizing subjective interpretation and human error. There are studies that have shown that AI-assisted analysis improves species discrimination, especially in complex or mixed samples, while it also enables faster and more consistent forensic decision-making. Despite the potential that we had learned, validation still remains one of the biggest challenges. So based on researchers, they emphasize problems such as PCR bias, contamination, bioinformatics mislabeling, and variation between laboratories. These factors can actually compromise reliability if standardization is not enforced. However, Forensic admissibility standards must be met for DNA metabarcoding to be accepted in the court. This also includes transparent methodology, known error rates, and validated protocols, which is also similar to other DNA-based forensic techniques. So, what do we need to look forward? For future developments, they include portable sequencing devices, which allows on-site DNA analysis and a real-time bioinformatics pipelines. The integration of AI and expanded forensic databases allows them to be faster in PMI estimation and also broader crime scene reconstruction. In conclusion, bioinformatics is the one that forms the backbone of DNA metabarcoding in forensic entomology. While challenges in validation and admissibility still remain, continued technological advances strongly support its future role as a reliable, automated, and court-admissible forensic tool. So that is all from my part. Thank you.